Hello, today I'm going to be demonstrating what is called a gram staining technique in microbiology. And what this is used for is to determine the difference between gram positive and gram negative. It's really important in medical establishments, for doctors, for patients all over the world, because knowing the difference, it can determine the doctor being able to tell you which type of infection, bacterial infection you have, and which type of treatment you should get, including medication. So you want to make sure, obviously, that you get that right the first time. There are a couple of steps, several steps to gram staining, and so I'm going to start and show you guys my materials. First and foremost, like any microbiology lab, gloves, we'll pretend to have gloves, they're the most important, and also goggles. So, in order to have the slide for the gram staining, you are going to want to put the bacteria on. So we'll say this is the solid bacteria and this is the liquid bacteria. The inoculating loop, Bunsen burner, striker, and then the Sharpie that I'm going to use to label the back of the slide. Make 2 circles. You never want to put more than 2. You can put it on separate slides. You can put it on the same slide. But if you put more than 2, it begins to look jumbled and it'll just be a big mess. So on the back, you'll put 2 circles. That way you know to put your small smear in there. And you will label them. And through the front, since it's clear and obviously going on a microscope, you will be able to see what they are while using the microscope. But the marker will not interact or interfere with the bacteria smear itself. So, what we're going to do next is, stick this there, bacteria smear. We're going to light the gas, turn the gas switch on, you should hear it, grab the striker, turn on the Bunsen burner, use the inoculating loop, run it over the Bunsen burner, get it hot, red hot, flame the tip, sterilize. Then you're going to want to grab the liquid. Make sure without contaminating the loop that you can take off the lid of the liquid bacteria. And so then you're going to put the lip of the bacteria just a couple of times over the Bunsen burner and then get your sample, put the bacteria lip back over the Bunsen burner a couple of times and replace the lid. Then you're going to want to make a very small, thin smear of the bacteria. And if you can barely see it, that's good. Like, you do not want to overdo it. Less is more in this case. Then you're going to run your inoculating loop back through the Bunsen burner, sterilize it, and in order to use a solid bacteria for the second circle on this slide, you have to have water. You have to mix it with water. And so you're going to put your inoculating loop over the sink. This is the imaginary sink over here. And you just put just a couple of drops to where you get like a thin film of water. And you're going to place that in the other circle. And then again, because water is not always completely pure, it's not, you're going to want to put the inoculating loop back over the Bunsen burner, and then you're ready to collect a small, barely dab the solid bacteria, and then put it on top of the water so that it mixes well and you're able to get a good bacteria smear. And like the first, less is more. You don't want a very thick slide. So after this is done, you will allow the water to dry by itself for 10 minutes or so. 10-15 minutes, you wait for it to go by, and then you will want to put the slide over the Bunsen burner. But you want to make sure that you don't burn it off. You just want to make it adhere to the slide, the bacteria. So you're just going to run it over it a couple of times, and then it should be good to go. So now we're ready for the gram stain. So the parts of the gram stain include the water, the primary stain crystal violet, which turns turns it purple. The iodine, which interacts with the primary crystal violet and will help the purple stay into the cell's walls and structure. And then you have the alcohol or decolorizer. And then lastly, your counterstain or safranin, which will turn gram-negative bacteria pink. We've got our rods, our staining tray, and then of course the heat fix bacteria slide or smearing slide. And then we have our napkin to dry it off when we're done. So first we're going to want to grab the primary, the crystal violet. This is going to turn all cells on the slide purple, gram-positive or gram-negative. First step, you get it really soaking wet. You let that sit for 1 minute. 1 minute goes by and then you rinse with the water and get it all off. And then you're going to want to use the iodine. The iodine, if it's gram-positive, will help set the crystallization and the purple inside the gram-positive cells structure. If it's gram-negative, the iodine will not set and the purple is obviously not going to stay for the next step. But if it is gram-positive, these two together, the primary and the iodine, will help. It'll start becoming crystallized. After that, you're only going to let the iodine set for about 15 to 20 seconds. And then you're going to rinse it as well with the water. The next step is probably the trickiest and one of the most important that you get it right. The decolorizer or the alcohol. After this step, if it is a gram-negative cell, it will be clear. It will have no color. If it is a gram-positive, it will remain crystallized in purple. So what you want to do, because you don't want to get this completely doused unlike the others, you just want to barely drip it on and until the color starts to run. You'll see the color running, and then you want to stop immediately, rinse it with water. And after that, you're going to use the counterstain safranin, which will turn it pink in color to better help see and view and, make data of. And so you're going to put this on there for a minute as well. And then you're going to rinse it off with water. So after you've got all of your things on there, you can remove the slide and place it on the napkin. And you're just barely going to want to dab. You don't want to lose and, have to restart all of the work you just did. So you just barely dab it to get it dry. And you allow it to air dry. And then after it's all dried up, you are ready to use a microscope.